let's take a look what a cell is. Okay, now I want you to look at the cell as if we're actually looking at what the coronavirus is trying to infect. Okay, remember the cell in the respiratory tract, the one that lines the respiratory tract, located in the alveoli, is made out of cells. And what the coronavirus is trying to do is go inside the cell. Okay, because remember the cell, the coronavirus does not have a cell, so it needs to go inside your body's cell in order to infect and produce copies of itself. And that's what this coronavirus is trying to do. So this is your typical cell, okay? And what do we have in the typical cell? We have the plasma membrane. So the plasma membrane is actually the cell's wall, okay? And then the plasma membrane is typically the covering of your cell. And the cytoplasm is the fluid component of your cell. And the nucleus is what you have located inside the cell. That's going to be here, okay? Which is made out of genetic material. And remember, that's the brain of your cell. That's the one that tells us what the cell has to do, right? So these are your structures that you see typically. The components, the basic components are the plasma membrane, cytoplasm, and nucleus. But there are other components that you would see also on a cell. The first one is we're going to try to see your plasma membrane, okay? So the plasma membrane is the extent of the cell. So it covers the cell, and it's made out of a membrane. It's made out of lipid bilayer, made of fats, okay? Made out of two lipid bilayer. So you can see that this is going to be the lipid bilayer summary. I'm sorry, I'm not getting my pen here. This is the first layer of lipids, or what we call fats. And then there's this other layer of the fat of the cell wall of the plasma membrane. So if you also take a look within the cell membrane, you got a lot of these proteins that are interspersed within the cell membrane. So these are your proteins, okay? So it's either made out of a protein with carbohydrates, and there's going to be a protein that is what we call a channel protein. And this channel protein is what we're going to be discussing in your physiology class, how things from the outside goes inside the cell, or if the things from the inside of the cell goes outside from the cell. And that's going to be in your physiology class. But just remember, this membrane proteins is what is very important, that signifies a receptor, okay? So the receptor, okay, the plasma membrane acts as a receptor because of these proteins. The cytoplasm has a jelly-like fluid inside called a cytosol, okay? And within that cytosol, you got a lot of the cellular elements that are suspended within the cytosol. And those are the elements that we're going to be studying one by one. But generally, the cytosol is made of water, ions, and enzymes, and also the cellular elements. Now, what are the cell elements we just start studying out from? The first one is called ribosomes. So within the cell, there's going to be this ribosomes. You can see those red dots looking at this violet structure. So those are called ribosomes, okay? So the ribosomes are really the one that produce proteins. Now, if you notice that antibiotics, okay, most antibiotics work by blocking bacterial protein synthesis. So bacteria also have ribosomes. But the antibiotics that we use, they're the one that allows the bacteria not to function and produce proteins. The virusomes usually are located around this purple structure. So this purple structure is called endoplasmic reticulum. Endoplasmic reticulum is like sacs, okay? And you see a lot of this ribosomes studied in the endoplasmic reticulum. So when you see a lot of these ribosomes studied in the endoplasmic reticulum, we call it the rough ER, rough endoplasmic reticulum. It means there's a lot of protein production here. But sometimes the endoplasmic reticulum are naked. They don't have ribosomes, so we call it smooth ER. Okay, so those are the different kinds of endoplasmic reticulum. Now, within the endoplasmic reticulum, you can see that ribosomes are located at the walls of endoplasmic reticulum. So the ribosomes keeps on producing these chains of proteins. So the chains of proteins is actually the one that we need in our body. So the ribosomes now travel to the sacs, and that is then packed in the endoplasmic reticulum. Okay? So when they're packed, they then are transferred to the Golgi apparatus. So if you take a look at these proteins that are packed by the ribosomes, by the endoplasmic reticulum, they're being transferred now into the Golgi apparatus. So the Golgi apparatus then allows it to be purified and packed again, and then they travel as vesicles. And then after that, they can be secreted outside your cell, similar to hormones, or this one is going to be passed on to another cellular element, and those proteins becomes enzymes called digestive enzymes, and then they're the one that will allow this body to produce what we call your lysosomes. Okay, so lysosomes are the one that will digest anything that enters in your cell. So that is what the Golgi apparatus is. They pack the product from the ER to move to the Golgi apparatus to be secreted outside your cell or to be utilized inside the cell. So that's your Golgi apparatus. Now, I've talked to you about a lysosome. Remember, a lysosome is a cell component that has digestive enzymes. Remember, this actually are proteins that were produced by the rough endoplasmic reticulum from the ribosomes. Now, this one, ribosomes are actually walled structures contain digestive enzymes. And the digestive enzymes are the one that will digest unwanted substances inside the cell. So it's just like a vacuum cleaner that anything that's not supposed to be inside the cell will be digested by the lysosomes. Peroxisomes, generally it's the same thing as the lysosomes, but they neutralize free radicals. Free radicals are actually chemicals that can produce cancer. So your body has a way to actually kill off those cancer-producing substances called free radicals. Now, the next one is going to be your mitochondria. So if you take a look at mitochondria, this is a typical mitochondria, okay? It has cristae, okay, and cavities, and these are the ones that produce what we call energy. Remember, the cell needs energy. Other things that are located in there are what we call cytoskeleton. It's a elaborate network of rods. Rods are proteins, and they're the one that creates the shape of the cell. So what are the cytoskeleton? We call it microtubules. Okay, so they're the one that creates the shape of the cell. You can see the shape of this cell. Okay, and microfilaments. Okay, they're the one that actually produce contractile filaments. Okay, in a cell, such as your contraction of the muscle, and then intermediate filaments. There are also what we call protein fibers to make the cell structure strong. So those are what you call cytoskeleton, and some other components. These are centrosomes and centrioles. Generally, they're the one that used for mitosis or cell, or what we call cell reproduction. Okay, and those are some of the cytoplasmic organelles. Now, there's another one called cytoplasmic inclusions. So these are temporary structures, but they're not present in all cell types. So they're temporary, and the main thing about them is they store substances such as lipid droplets in fat tissues, okay, or glycosomes. They're the stored form of sugar called glycogen. So those are what we call cytoplasmic inclusions. So these are general structures that you see in the cell, but the main structure in a cell is the nucleus, okay? So the nucleus is the brain of the cell. The DNA inside this nucleus is the one that directs the cell's activities, okay? So it is like the brain of the cell. And it has the DNA, which is considered a central core or the control center of the cell. It has a nuclear envelope, a covering on its DNA on the nucleus called the nuclear envelope. And within that area of the nucleus are strands of chromatin materials, chromatin. And then in the middle is called nucleolus. It's called also a little nucleus. And the main function of this is the production of ribosomes. Okay, so this is what you see on a cell. So generally, you have the plasma membrane, the cytoplasm, and the nucleus. Now, the microscope's purpose is to magnify. Okay, what is magnified? Make it bigger. It is to resolve, okay, power. It has a resolving power, meaning make it clear, and it has contrast. It has light and dark. The microscope has what we call an ocular lens. So the ocular lens is the lens that you see where you put your eye on to visualize, okay? And that is going to be the eyepiece that you see here, okay? So we have binocular, okay, eyepiece. That is where the ocular lens is, so we call it an ocular lens. Okay, and remember the magnification generally of the ocular lens is 10 times. So I'm looking at a specimen just by the ocular lens, it's being magnified 10 times. Remember, the number is very important because I'm going to be asking you for magnification power of the microscope from the combination of the ocular lens and the objective lens. So just remember, ocular lens 10 times. Now the arm, okay, serves as a handle. And then the nose piece is a dial that you see that is just located below the ocular lens. And this dial has objective lenses here. Now, below the nose piece are objective lens. Okay, so the lower lens is attached to the nose piece, and the magnification of each is stamped on the housing. It actually tells you what is its magnification. Okay, and that's actually very important to know so that we can know how the specimen is being further magnified. And low power objective is what we call scanning objective. So whenever you start looking at a microscope, always use scanning objective or what we call low power. Low power has this, if you take a look at the magnification of that, it is actually 4x, 4 times magnification. Okay, now why is that important? Because whenever you put a slide underneath the low power, you put a slide here underneath the low power, so you're trying to magnify the slide not 4 times, but you have to multiply the 10x of the ocular lens. Then multiply it by the objective lens, which is 4x. So the total magnification when you utilize the scanning objective, okay, is going to be 40x. What is 40x? So it means that whatever you see on the slide, on the naked eye, in a microscope with this setup, ocular lens and low power, you're looking at a specimen 40 times its size. Okay. Now the next one is called medium power objective. So once you finish looking at a specimen and it's clear, then you can move on to the next objective, which is going to be higher magnification, and it is called medium power. Now, if you take a look at medium power, that is 10x. So total magnification equals 10 ocular lens times 10 objective lens. So total magnification is 100x. So what is 100x? You're magnifying that the structure that you see on the slide 100 times its normal size. So high power objective, the magnification we use is the 40x. Okay, so the 40x is the high magnification. So 40x is the objective lens. So if I use total magnification, I'm going to use the ocular lens magnification 10 and then multiply it by 40. So my total magnification here is 40x. So that is meaning that whatever you see in the slide is 400 times its size. Okay, the stage is usually where we put the slide in, okay? We put a slide here underneath the objective lens so we can visually look at what the slide has. So that is the stage. And remember, the stage have clips on each side. So when you put the slide, let it be captured by the clips because the stage can move from up and down and left and right. So that is the reason why the has to be put on those clips. Now, the next one is going to be the illumination. So this is a form of illumination with a microscope, and this is where the light comes out from. So light goes into the slide so you can actually visually see it on the microscope. That's called the illuminator or the light source. And there's other things located there underneath the stage. You have the diaphragm, either it's diaphragm lever and the diaphragm ring. So they're located underneath the stage. And generally, the purpose of this is just to regulate the amount of light entering the microscope. So you can literally adjust the amount of light entering the microscope. You can spread it wide or you can focus on one area. And that is actually the iris diaphragm. And then we have the condenser. So condenser is a lens that is found beneath the stage, and that is used to focus the light on the specimen. So a condenser is the one that you focus onto the bottom of the stage. So this is your condenser. Okay, and then on the side, we have the coarse adjustment. So the coarse adjustment, okay, is a big dial. And remember, when you turn the coarse adjustment, it will actually make the stage go up and down. Coarse adjustment get really big steps of putting it up and putting it down. But the fine adjustment, is the small dial, that is the one that will actually finely tune it to go slowly up and down. So fine adjustment is very important. Fine adjustment is usually can only be used on high and medium power objective, but on the scanning objective, the low power objective, you can use your coarse adjustment, but never use coarse adjustment when you are on the middle and high power objective. Okay, and then the base, as I said, is way to support the microscope, and that is for proper handling of the microscope. So we studied about the parts of the microscope, we studied about its function, and we studied about how the care of the microscope in, okay? And the next topic is principles about microscopy. So on focusing the letter E, so we have a slide that has a letter E, okay? So we're going to be looking at that slide inside the microscope, okay? So the first thing is lower the stage of the microscope using the coarse adjustment, and then place the slide of the letter E in the stage, okay? And stabilize it with the stage clip, okay? And then after that, I ask you to look at the letter E in the center of the slide, okay? So now the lowest objective is going to be used. So you're going to use scanning objective. So whenever I say scanning objective, that's low power objective, okay? So look into the eyepiece, okay? And slowly use the coarse adjustment. So at this point, since you're using the lower objective lens, you can use the coarse adjustment. But if you go higher, the medium and high power objective, you can never use the coarse adjustment. So use the coarse adjustment, and then when it becomes clear, okay, try to use the fine adjustment, okay? So once you see the letter in the microscope, okay, now you can increase or decrease the contrast, which is going to be trying to see the amount of light, okay? So on the bottom of the slide, you can turn that lever and you can see that sometimes you can spread the light out, or sometimes you can actually focus the light on. So you just spread the light out so you can see the letter E. And then from there, do the fine adjustment again to sharpen the focus, okay? So that's how you use a microscope, right? Now, in the microscope, okay, looking at the letter E in the microscope, okay, using the total magnification of 40x, okay? So you use the total magnification of 40x, and that is the ocular lens 10x plus your ocular lens, okay? So 10x and 4x, they'll give you the total magnification of 40x. So when I look at the microscope, remember, take a look at the picture on the right upper hand of the slide. It is the letter E in upright position. So when I look at this specimen in the low power objective at 40x, you can actually see that the letter E is inverted and it's upside down and reversed. Okay, so that's how you see it on the microscope. Even if you see it on the upper right hand, you can actually see it. It is upright. Okay, when you see it on the naked eye, it's upright. But on the microscope, what happened is it is inverted and reversed. Okay, so that's how you see the letter E. That's how what you see on the letter E. Okay, so and also if you see the letter E in 40x total magnification, you can see the whole letter E in the microscope. Okay, and the field. This is called the visual field. The circular field of view. Okay, we call it a field of view. Okay, is what we see in a microscope. So this is what it looks like on 40x magnification. The whole letter E is visible inside the field of view. Okay. Now moving on to the next one. I'm going to use the medium power objective. So the total magnification is going to be, so medium power is 10x, ocular lens or lens objective, ocular lens objective is 10x, total magnification 100x. So if I look at it in a microscope, it's still going to be upside down and reverse, but I can only see small pieces of that letter E. Okay, I'm not seeing the whole letter E, so I'm going to see only a small fragment of that letter E there. So the field of view, okay, here, the field of view, okay, is lesser, meaning it's magnified. Okay, so again, going back to the first one, the field of view, okay, is actually bigger, okay, because I can see the whole letter E. But here, the field of view is now going to be lesser because smaller is because I can only see small parts of the letter E, but it is more magnified, okay? So this is what it means about field of view, right? Now, moving on to the next one, I'm going to go to the 40x magnification that it's supposed to be high power, okay? So this is high power magnification. Now, in high power magnification, total magnification is 40x. So if I look at a microscope, it's still going to be upside down and reversed. But I can see lesser of the field of view, but it is more magnified. Okay, so it's getting more magnified, so the field of view becomes less. Okay, so that is what you see on the high power magnification. So high power 40x. Okay, field of view is lesser, but it is more magnified. All right. So what happens to the microscope image when you move the microscope slide towards you? So if you're looking at the microscope, okay, and this is the letter E. So letter E is upside down in reverse, okay? So while you're looking at a microscope, okay, if you move the microscope towards you, okay, what's going to happen on the image when you see it on a microscope? Remember, it's upside down in reverse, so it's opposite. So the image will move away from you, okay? That means away from me. So if I move this microscope towards me, the image will move away from me, okay? So what happens to the image when you move the microscope slide away from you? So now if I move the microscope slide away from me, okay, remember this is here, that is you, me. Now the image moves towards me, okay? So because it is upside down reverse, okay? Now another question is, what do you move your eyepiece image? How do you move your eyepiece image to the right? So I want to see the microscope image. So this is what I see in a microscope, upside down reverse, okay? I want to move it towards here, okay? towards here. I want to see that image that will actually go towards to the right. So I want to see that image towards to the right. So the way to do it is to move the stage to the left. Okay, to move the stage to the left. So because remember, it is reverse. Okay, so this is what you see on the microscope, right? So that finished up our lab about the microscope, on how we look at the image on different magnification and moving the image of the slide in the stage compared to what you see on the slide. Okay. All right, so working distance is the distance between the objective lens and the specimen. So if this is your objective lens, okay, and this is the lens, objective lens, let's say this is 4x, okay, and this is your slide that you put on the stage. What it means by working distance is the distance between the lens and the slide, okay? This is called your working distance. Okay, so generally on the lab, I'm going to be asking you to take a look at the working distance between 4x and the slide. Okay, and see that letter E is clear. Okay, and I'm going to ask you to see the objective lens. That is going to be the medium power, which is 10x, and then the slide, okay? So 10x and the slide, and then see the letter E, now it's clear. So the working distance between the medium power, 10x, will actually becomes lesser, right? The working distance, okay? Now, if I ask you to move it to the high power, okay, this is the objective lens, and the slide is here. And when I ask you to visually look at the letter E, and now it's clear, the working distance is lesser. Okay, so that is what's happening into the different working distance of the different magnification field. Okay, lesser working distance. Okay, so that means that you really have to be careful when you use high and medium power objective. You have to use only fine adjustment. You only have to use the fine adjustment. Why? Because if you use the coarse adjustment, you can break the lens or break the slide, okay? Because the working distance is lesser when you keep on going to the higher magnification, okay? So that is what working distance is, okay? But generally, the working distance decreases, okay, in your high power, but the magnification increases. You're seeing a bigger sample of the specimen. So that is the concept of microscopy, okay? Now, the circular field, okay, the circular field that you see in the microscope, okay? So let me erase my slides. Now, this is what you see in the microscope, okay? This area on the circular view, whatever you see inside that ocular lens, this is called the field of view. That's called the field of view, okay? So the field of view changes at different magnification, right? Remember the field of view of 10x, you can see the whole E. I mean 4x, scanning objective. At 10x, you can only see a portion of that letter E, okay? And then after that, at 40x, high magnification, you can probably just see a segment of that letter E, and it's more magnified, okay? You can only see a short part of that letter E, okay? So the size of the specimen can be estimated of the diameter field of view at each magnification. So I want to see what is the field of view distance. So I want to measure how many micrometers is this field of view. Let's say 0.1 micrometers. So I see a transparent ruler that was placed onto the slide, and this tells me about the field of view of the area. Okay, so remember, this is 1 millimeter. Okay, that's 1 millimeter. Add another millimeter, okay, right here. So that's 2. And then this is another millimeter, that's 3. And then this is just about 3/4 of a millimeter. So let's say this is going to be 3.75 millimeters. Okay, so this is sometimes the way that we measure it by using that transparent ruler. Okay, so now let's make sure that we know that we usually use micrometer instead of millimeter. Remember, 1 millimeter is equivalent to 1,000 micrometer. Let's say this is going to be 4 millimeter. So the field of view is 4 millimeter, but instead of saying millimeter, we're going to use micrometer. So convert millimeter into micrometer, and the answer is the field of view here is 4 millimeter, sorry, here, but it's also equivalent to 4,000 micrometer. So that is another way to measure, okay? So the field of view of 40x is 4 millimeter, but we use the word micrometer. So it's actually 4,000 micrometer in size, okay? So that's the way to actually look at it when we go and measure the specimen by using a transparent ruler. Okay, so using this as a comparison between different magnification. Okay, so a 40x, okay, a 40x is about 4,000 micrometer. Okay, and then 100x, I only see 1 millimeter, which is 1,000 micrometer, and about half of that millimeter, which is 500 micrometers. So it's 1,500 micrometers. Okay, but in this case, in 400x, I generally see just really about 3/4 of that millimeter, about 75.75 millimeter. Okay, so that will be equivalent to 750 micrometers. Okay, so the more magnified field that you have, the field of view becomes smaller, okay, but the specimen becomes magnified. So that's how you study about field of view. Okay, so now let's take a look at the concept, okay, and try to finish this one. What is the highest possible magnification that can be obtained when using this microscope? The possible highest magnification is going to be objective times ocular, and the answer is going to be the letter D, 40x for the objective, but we're going to use the ocular 10x and 40x, so the total magnification, the highest is 400x. So that's the highest magnification from the microscope. What happens to the amount of light in the field of view when switching from low to high? So what you're going to be observing is when you look at the specimen on the low objective and suddenly you change to high objective, the amount of light actually decreases, okay? So it's going to be darker. So that field of view gets darker. So that's the reason why you got to put the amount of light, increase the amount of light so you can see the specimen. So again, that's the concept of changing from low power to high power objective. The amount of light becomes lesser, okay? So what happens to the field of view when switching from low to high? The field of view, okay? The field of view is the one you see on the circular view, okay? So what happens to the field of view from low to high power? The field of view decreases, okay? Decreases. Remember, we measured the field of view from low power to high power. It started at 4 millimeter, then it becomes like 0.75 millimeter. The field of view decreases, so you see less of the slide, okay? from low to high power. The field of view decreases. You see less of the slide, but you magnify the specimen. This is what you see on the slide on the naked eye on the slide, the letter F. But which of these four you see on the low power objective on the field of view? Would it be 1, 2, 3, or 4?
2.
Okay, so it would be 2. Inverted and reverse. Okay, inverted and reverse. Okay, so that is exactly how you see it on the microscope. Okay, very good. Okay, now this image becomes inverted and upside down, what you see on the microscope. Okay, and then on 40x, this is how you see it. Compared to 100x, you only see the field of view becomes lesser and a specimen becomes enlarged or magnified. Okay, so objective appears larger, field of view becomes smaller. We have an answer to this one. The total magnification of an image is the result of the combined magnification of the blank. Which is the answer? Did we ever say eyepiece? We didn't even say an eyepiece. And also diaphragm. Okay, so you eliminate that one. Okay, so this is how you answer questions. You actually really do elimination. So this doesn't sound good. Okay, so the answer is objective times eyepiece. Okay, so eyepiece is actually really ocular lens. Okay, ocular lens. So that's actually the best thing because here objective and mirror. Okay, we didn't use mirror. Here, low power and high power, we didn't actually use that, okay? So the closest thing to the answer is going to be objective eyepiece. Even though we didn't use eyepiece, it is still the ocular lens. Let's say the field view is 1 millimeter, which is 1,000 micrometer, okay? Can you tell me the size of this organism? What's the length in micrometer? We use 1 millimeter. They just call it 1,000 micron. So the field of view is 1.5 millimeter or 1,500 micrometer. There are 3 cells in field of view. So you have 1,500 divided by 3. So each cell is 500 micrometer. So if you want to see the total magnification, just multiply ocular lens. Remember, ocular lens is always, and it's on our microscope, is always 10x. And then use the objective lens, okay? Our objective lens is usually what we call 4, 10, and 40, okay? Those are the 3 objective lenses that we have. So if you want a total magnification of the low power or scanning objective, that's 40x. And for medium power, that is 100x. And for high power, that's 400x. This is what a micron looks like on a dot, and this is the diameter of a human hair. That's how small a micron is, okay? Again, this is a micron, a small dot here, and this is a very small drop of water, 15 microns. There's a little bit bigger drop of water, 30 microns, and this is a human hair that's 80 microns. So microns is really a small unit, okay? Very small, okay? And that ends up our microscope.